Hello, we shall be discussing on the DNA packaging through a nucleosome concept. In a non-dividing eukaryotic cells, the DNA is associated with a basic protein called as the histone proteins that form the deoxyribonucleoprotein or the chromatin fibers. These chromatin fibers they form a bead-like appearances. So the appearance of these chromatins appears bead on strings consisting of 8 histone proteins. So when we observe in the electron microscope, the chromatins along the length of the chromatin fibers, that is consisting of DNA, histone proteins occur in the beaded form. So it is due to these appearances of histone proteins forming a bead along the length of the chromatin fibers. The chromatin fiber shows a bead of string-like appearance. Histone proteins are basic proteins having high percentage of basic amino acid, that is arginine and lysine. These histone proteins are of 5 different types in this DNA packaging. The 5 different types are H2A, H2B, H3, H4 and H1. And out of these 5, the first 4 histone protein, that is H2A, H2B, H3 and H4, they occur in pair. So when these 4 occur in pair, they form 8 histone protein. So these 8 histone protein are also called as the histone octamer. So around these histone octamers, they form what is called as the core particle or the new body. So these new body or the core particles are formed by the histone octamers. Now along the length of this core particle or new body, around 146 nucleotide of DNA form 2 circles. So these 2 circles of the DNA along with the histone octamer, they form what is called as the nucleosomes. And this nucleosome have a diameter of around 11 nanometer. And this one nucleosome is attached to the adjacent nucleosome through a linker DNA or the interbead DNA. Now the remaining, that is H1 histone, is tissue specific and is present once per around 200 base pairs of the DNA and is loosely associated with this DNA. So this nucleosome together with the DNA, when it is observed in the electron microscope, they observe in this form. So this is the nucleosome consisting of the histone octamer and the 2 circles of DNA, and this is the DNA segments, and these are the linker DNA or the interbead DNA. So due to this appearance, the chromatins give a bead on a string appearance. So this is all about nucleosome concept.